Today I'm going to talk to you about the process of the big three. The big three includes replication, transcription, and translation.
Replication transfers from DNA to DNA. In molecules of DNA from one original DNA molecule, replication occurs in all living organisms, acting as the most essential part for biological inheritance. This is essential for cell division during growth and repair of damaged tissues, while it also ensures that each of the new cells receives its own copy of the DNA.
The cell possesses a distinctive property of division, which makes replication of DNA essential. Replication is also known as a semi-conservative replication. Within each fork, one DNA strand, called the leading strand, is replicated continuously in the same direction as the moving fork, while the other, lagging strand, is replicated in the opposite direction in the form of a short Owazaki fragment. It reads from 5' to 3'.
The helicase is the enzyme that unzips the DNA molecule. They separate each double-stranded into single strands, allowing each strand to be copied.
One of the key molecules of DNA replication is the enzyme, DNA polymerase. DNA polymerases are responsible for synthesizing DNA. They add nucleotides one by one to the growing DNA chain, incorporating only those that are complementary to the templates.
Okazaki fragments are short sequences of DNA nucleotides, which are synthesized discontinuously and later linked together by the enzyme DNA ligase to create the lagging strand during DNA replication.
Primase is the enzyme considered as a bookmark, which shows polymerase where to begin a fragment.
And lastly, the ligase sews together the fragments of both the leading strand and the lagging strand.
Transcription. Transcription is the process of DNA to mRNA. Transcription is the process by which the information in a strand of DNA is copied into a new molecule of messenger RNA, mRNA. The sent strand is a strand of DNA that has the same sequence as the mRNA, which takes the antisense strand as its template during transcription. Transcription also reads from 5' to 3'. In transcription, there is a specific bubble for each protein, and it uses uracil as a base. The helicase in transcription is basically the same as other. It's the enzyme that unzips the DNA molecule. The RNA polymerase is the enzyme that promotes base pairing. This uses uracil as a base, and it's also read from 5' to 3'. Lastly, we have translation. Translation is also known as protein synthesis, which is mRNA to tRNA to amino acid change, and then finally to protein. Translation is the process in which ribosomes in the cytoplasm or endoplasmic reticulum synthesize proteins after the process of transcription of DNA to RNA in the cell's nucleus. It's an mRNA strand, and it reads from 5' to 3'. Codons in the mRNA are read during translation, beginning with the start codon and continuing until a stop codon is reached. They are grouped together in numbers of 3. The ribosome has a small and large subunit, and its complex molecule composed of several ribosomal RNA molecules and a number of proteins. APE is a small section of the ribosome which is split into three parts: A, attachment site; P, polypeptide chain; E, exit site. APE includes 20 amino acids and contains 60 codes. The anticodon sequence determines the amino acid that the tRNA carries. tRNAs function as a specific site in the ribosome during translation, which is a process that synthesizes a protein from an mRNA molecule. When a tRNA recognizes the binds to its corresponding codon in the ribosome, the tRNA transfers the appropriate amino acid to the end of the growing amino acid chain.